Thank you. Our next presentation is SmartCella, and I'd like to welcome Nina Bauer, Chief Business Officer, to the stage.
Thank you very much for the introduction and welcome. Thank you for joining us. And it's particularly nice to see so many familiar faces in the audience. My name is Nina Bauer. I'm the Chief Business Officer of SmartCella. And today I will be talking about two topics. The first one is our lead program, which is SMART-01. It's a cell therapy for heart failure. And the second one is the extraducer, which is a medical device that enables the delivery of a wide range of payloads directly to the target tissue. SmartCella is a Swedish company, and we focus on two key areas. One are the delivery solutions, and the other one is therapy development. Our delivery solutions are the extraducer that I've already mentioned and that I will be talking about today. And we also have a cell-mediated platform. And on the therapeutic side, we have mRNA-based therapies and stem cell-based therapies for tissue repair or tissue replacement, and I will focus on that today. The company was founded about 10 years ago. We are still based in Sweden. Most of the technologies that we have are spin-outs out of the Karolinska Institute. Today we have about 70 employees, and we have raised more than 100 million euros over these 10 years. Our vision is really that by combining first-in-class delivery technologies with the novel therapies that we're all working on, we can really make a difference, and we can really unleash, we like to say, the full potential of the therapies that we're all working on and why we're all here. And I think actually that we will be able to solve some of the problems that we've been talking about all week, which is we still can't really make enough of the drug product. We make it, but it's expensive. We can't get to where it needs to be, and that includes solid tumors. So all of these topics are recurring, we're all talking about them, and I want to show you a little bit of how we are approaching these and hopefully how we can maybe work together and help with overcoming this. We currently have 3 programs in development. I will be focusing on the first one, which will be entering phase 1/2A next year. And I'm going to start with that. So heart failure, or to put it a lot more complicated, advanced ischemic heart failure with reduced ejection fraction. I'm going to call that heart failure because this is a tongue twister that I can't navigate very easily. But in essence, a patient who is part of this diagnosis goes through a progression that you see here on this slide. Typically, we see a clinical onset of some form, some symptoms manifesting, and we can control that relatively easily or routinely with small molecules. Over time, the symptoms worsen. We see a hospitalization. In some cases, a device therapy can help, which means typically implantable defibrillators and similar. But inevitably, there will be a progression. We will see frequent hospitalizations all the way down to pump failure. And this is really the patient bracket where we see very limited therapeutic options. So logically, that's what we will be focusing on. So our focus population represents about 5 to 10% of the patients that fall into this heart failure group. They typically have a very high burden of disease, which means they have significant morbidity, significant mortality, and most importantly, treating these patients is extremely expensive. The therapeutic options, as I've mentioned, are relatively limited. And also, if you look at the drug pipeline, there's not that much out there. So the way we are planning to address this is with an allogeneic cell therapy. I mentioned this is a technology out of the Karolinska Institute, and it is human ES-based, so human embryonic stem cell-based. The differentiation protocol is such that we go all the way to ventricular progenitor cells. They're characterized by ISL1 expression. And then we purify them by removing the Tra160 positive cells. After that, they can be transplanted, as you see in these pictures on the right. So what does this look like? This is GLP data. This has been published, but we use mini pigs for this. Pigs are suitable models for heart indications in general. Their hearts are very similar to humans. That's why we see xenotransplants in some cases where people actually transplant pig hearts. But bottom line is it's a great model for us. And what you see here in white is the infarcted zone. And I'm going to try and point to that. No, that's not working. Yeah, so white is the infarcted zone, and in red you see our grafts directly after transplant. And then after 3 months on the right here, you see that the cells actually fully differentiate. They connect with the host tissue and they vascularize. So they really become part of the host tissue and integrate well. From a functionality perspective, what we have here is both the ejection fraction data and the infarct volume. This means these pigs were infarcted 2 weeks prior to transplant. A day before, we did all sorts of measurements and assessments, and then what you see here as the 0 time point is when the cells were injected, and then we have monitored these animals for 3 months. So in orange, you see the treatment group, in brown is the vehicle group. And from an injection fraction, as I said, you have the infarct, and then 2 weeks later, you measure the time-0 time point. And if these animals are untreated, you typically see a continuing decrease, which is, as I said, evident here in the vehicle group. In the treatment group, we have a non-significant data point, which means we haven't seen any further decrease. We could stabilize these animals from an ejection fraction perspective. Conversely, on the infarct volume side of things, the infarct 2 weeks into the model, you have a relatively stable infarct zone, and that doesn't change much. It doesn't become better, it doesn't become worse, it remains stable, which is what you see here with the non-significant data. On the other hand, in the transplant group, you actually see a decrease in infarct volume. This means we have a certain level of regeneration in these animals. From a functionality perspective, what you typically see and what makes sense is you see a level of arrhythmias. And this is, you have a highly functioning organ, lots of electricity going around in these coupled cells, lots of muscle activity, and so you're injecting cells into this, you typically see arrhythmias, or engraftment arrhythmias as they're called. And so we observe that here. We have 3 groups, 3 treatment groups with different cell numbers that were transplanted, and in all groups we see both the heart rate go up and we see the arrhythmias going up, but we see them going down after about a month. That is faster than what we have seen in comparable studies. And most importantly, we have also seen no animal death. So while we have the typical observation of these arrhythmias, they come back down, these animals stabilize quite nicely. So this program, as I mentioned, is going to go into the clinic. It was until recently partnered with AstraZeneca. They have now become an investor and have externalized the development back to us. We have our facility set up. It has passed inspection by the Swedish authorities at the beginning of, well, actually just before Christmas. It was a nice Christmas present. And we're in the process of finalizing our data packages, which are going to be submitted in the coming months, and then we're planning on entering the clinic middle of next year. Since AstraZeneca are only an investor at this point, we're also looking for partners. So before I let you go, I want to talk a little bit about the delivery, but because these patients that we're treating, as I mentioned at the beginning, are highly fragile, right? These are patients at essentially end stage, and the last thing you want to do is an open heart surgery where you inject cells into the heart. And so what we're leveraging here is our second technology, which is the extraducer. It's an endovascular catheter, and we can use this for minimally invasive cell delivery in this case. The way it works is it is a so-called inverted Seldinger technique. So when you use any kind of catheterization or injection into the bloodstream where you have blood drawn, that is the Seldinger technique. It was discovered and developed at the Karolinska Institute, and here we're going inverted, i.e. we're going into the vessel, we navigate to where we want to be, and then we exit the vessel on the merit or merited by this very, very sharp needle tip. This means that we have very limited to no hemorrhage and thrombosis risk, and we can really go to where we need to be and deposit payloads into the target tissue. These payloads can be anything from small molecules all the way to cells. In this case that I'm talking about today, obviously we transplanted the cells. It's relatively low tech, so it's not that complicated. We have a very long needle, which is this part in the middle here. I can't get this to work. Anyway, the middle part is the infusion needle. It's a very, very long, thin metal tube that has a proprietary way of sharpening it. Then we have all of these hubs that are hooked on their lure locks. All of this is pretty standard so that it can interact with accessories like syringes and things like that and just function pretty standard. What makes it special as well is the outer protective sheath. So because this is so long and thin, we protect it with a very thin plastic sheath that also avoids that this needle pokes where it's not supposed to poke until we are where we need to be. So what we have done in the case of our animals is we have done the same thing as before. We have infarcted the cells. We have taken post-transplant data using MRI, which is what you see on the right. And then the colorful circles that you can see here is where we planned to do the implants, the injections. So this is pretty standard software that you can find in any hospital that has imaging equipment. And I'm going to try and rotate this around. So here you can see very nicely in red is the infarct zone, blue is the rest of the heart, and then you see the colorful circles, which is where the injection is going to be planned. And in the heart, this then looks like this. What you see here, this long, thin gray shadow, that is the catheter. And then I'm going to start this video, and you can see the heart rotate, you can see it beating, and you can see the catheter operating there, navigating to this purple circle at the bottom. So to conclude with, we are very much looking for new partners, both for the cardiac program, but also the catheter is open for collaboration with anyone. So if you have a payload that you need to get to anywhere but the liver, come to us, we can help with that. We have achieved 510K with the FDA for abdominal organs. That means anything except for the heart. The heart makes this a class 3 device, which means that you will reference our device master file that is with the FDA in order to go into the clinic. And we will be starting 3 clinical trials with the catheter ourselves. One is the heart program that you have heard about. Another one is pancreatic cancer treatment with a chemotherapy delivered directly into the tumor to see if we can make a difference to IV infusion and systemic treatment. And the third one is our first partnership, which is a gene therapy for cardiac repair. And with that, thank you for your attention.